I'm going to talk to you about some of the things that are really going to change the way that our health service operates, and it's going to happen very fast. And there's lots of ethical and social issues that we need to consider behind these as well. My own background is I'm Professor of Molecular Pathology here, which means I'm not a geneticist, but I like to know what genes are doing in somatic tissues. So my main interest is cancer research, in particular thyroid cancer research. And really, what we want to do with your genes is be able to identify better ways of treating you with drugs, understanding what your prognosis is likely to be, so we can better advise you on the options that you have going forward with whatever disease you have, and also a better way of diagnosing people. So it really helps when you're ill if you've got a label you can pin on your disease. And we are starting to unravel the whole genetic concept of different phenotypes, so what your disease looks like, can actually be governed by different genotypes. So it's an exciting time to be involved in all of this, but it is complicated. So let me see if I can do the technology. I'm not very good on technology. Well, I always realize how old I am when I put slides up like this. I wasn't born when Watson and Crick discovered or identified the structure of DNA, but I spent a lot of time in Cambridge where you can't avoid that particular structure because it seems to be all over the place. And it's amazing to think how far we've traveled in 60 years. So we started off with the structure of DNA in the 1950s, and in the 1960s, we were able to identify the genetic code, and then as we move forward, we start then thinking, oh, this is good. Perhaps if we understood it better, it might be able to help us. And we have things like the polymerase chain reaction that we use so much in many of the techniques that we use. My students think I'm ancient. They think I was born in the Victorian ages when I say, well, when I started working, the only way you could do that was to move it from solution A to solution B at different temperatures and things like that. And they go, there's a machine. You just press the button. And it's true, technology does move very fast, and I start to feel more and more like a dinosaur as time goes on. Then, of course, okay, so we've got the technology. Well, let's sequence the whole genome. And this is where you really start to realize how much quicker we move these days. From deciding we were going to sequence the genome to actually getting the full sequence was nearly 11 years. Now we do that overnight in a machine, in fact, even less than overnight. So it's really quite scary how fast this technology is taking off. It's fantastically powerful. It involves masses of computing power, but it has the potential to unlock all sorts of different treatment options for us going forwards. So this is what we hope the technology is going to do. My favorite subject is pathology. I like looking down microscopes. I am weird, I must admit, but I do like looking down microscopes. But actually, that only tells me what my eye can see. Hidden in that is a massive amount of information that when we use genomic pathology, we can start to tease out why that looks the way it does. And we can use the individual things we find from this discovery to have biomarkers which enable us to predict the likelihood that somebody is going to respond to a particular therapy, to understand where the disease has come from in the first place, and also to predict whether somebody is likely to get some form of disease in the future. So it's tremendously powerful technology. And the idea, of course, is to turn pathologists from this, and my experience, most pathologists feel like this most of the time, to one of these. And if anybody finds any pathologists that look like that with a smile on their face, please tell me, because I want to employ them. But we really are starting to get into a very exciting field where we're not constrained by what we see down the microscope. We still need to be able to see what we see down the microscope, and we still need people to interpret that. But we can add so much more to it now. So what is genomic medicine? Well, this is the definition that you'll find if you search for it on the internet. It's an emerging medical discipline, well, I think it already has emerged, actually, that involves using genomic information about an individual as part of their clinical care for diagnostic or therapeutic decision-making. and the other implications of that clinical use. I love the way they always have something at the end of the sentence that makes you think, hang on, I don't really understand it at all now. So basically, we define genomic medicine as involving either DNA or RNA. And for those of you who are not au fait with all of this, your DNA is in the nucleus of your cells. It provides information to the rest of the cell in the form of messenger RNA, and the key there is in the name, it's a messenger, to go and make proteins, and the proteins are what actually brings your cellular function together. And the proteins are the drug targets, but how they're coded for is locked up in the DNA molecules. There are different subtypes of DNA, and there's the germline, which is the DNA sequence you inherit from both of your parents, which is a mixture of your parents' genes. Mitochondrial DNA you only inherit from your mother. So women, of course, are much better than men because we have 2 bites at this cherry. And you'll remember the information that was in the press recently about some mitochondrial DNA technologies whereby you can actually use the mitochondrial DNA from another female and put it into another female's eggs. Controversial, but for people who suffer from diseases caused by their mitochondrial DNA, extremely important. And you have somatic DNA, which is my personal favorite, because this is the DNA that when it's changed, it's normally the same as your germline in your normal tissues, but when it's changed, like in diseases like cancer, that's when it gives you problems. Okay? So you've got 3 different types there. Most people worry about their germline DNA more than anything else, because obviously if you find changes in that sequence, you're likely to pass those on to your children. But you'll also have inherited them from your grandparents. So where do doctors get your DNA from? Well, if we want to look at your germline DNA, the DNA you've inherited from your parents, it's very easy, actually. We can get a nice sample of blood from you, providing you're not needle phobic, of course. But we can get a nice sample of blood from you fairly easily. In that blood, there's lots of different types of cells. And the cells that we're interested in are called the white blood cells. And they have nuclei. And we can do very simple, it's a bit like cooking, really. It's not, don't let these molecular biologists tell you it's difficult. It's not. If you can follow a recipe, you can do this. And it's very easy to extract that DNA. So that will tell us what you've inherited. What we're most interested often in, particularly in cancer, but in many other forms of disease, is what has happened to that DNA, how it's changed in disease. And that's your somatic DNA. And you can get this either from the blocks that we use in pathology to diagnose your disease. These are fixed in formalin to stabilize them and then put into paraffin so we can slice them more easily to make our sections to look at down the microscope. Or you can get it from fresh or frozen pieces of tissue, say from an operation. What you then do is extract it using, and these are just simple kits. It is literally like following a recipe. And this is what your DNA looks like when you extract it. You see those thin sort of filaments in that solution? And that's the DNA being precipitated. And of course, your DNA is a structure like this, the double helix, that is simply got 4 different bases in it. And everything about us comes from the sequence of those 4 different bases. Quite amazing when you think about it, really. Now, the DNA codes for the messenger RNA to make the proteins. And the way it does this is it has different sections to it. It has an exon, which is the bit that codes for the protein, but it also has masses of DNA in between. It's not quite to scale, as you will see, called the introns. And those bits don't code for the proteins. They get cut out when the protein is made. And you can have various orders of these. So this one's a long one, and these will code for different proteins. Okay? The changes in the DNA code can lead to changes in protein structure, and that's usually what causes the disease. But only about 1% of your entire genome encodes the proteins, and that's called the exome. So there's an awful lot of bits in between that we actually really don't understand, and we call that dark matter. It's not translated into proteins, but actually, we do know now, and it's only over the last few years we've really understood this, that some of those regions that aren't translated into proteins actually form other complexes, not protein-based ones, but RNA-based ones, that regulate the function of the genes. So it's a lot more complicated than we thought. So we have the sequence, but there's an awful lot more that we have to learn about how that sequence actually makes proteins and controls the functions of the cells. So the good news is I'm going to be in a job for quite a long time. Now, we already use genomic medicine. We probably don't think of it as that, but actually it's not new. It's like most things in science. Most things in science are not new. They're usually reinventions of something that was there before. But we usually only look at single genes. So this is just a cartoon of the BRCA gene, which codes for breast cancer when it's mutated. And mutated means the sequence has just changed. And you can see that this, if it's mutated in your germline, has a considerable effect on your risk, depending on which type of the gene you inherit, of getting breast cancer. And of course, those genes also affect your risk of getting ovarian cancer. So one gene, which is actually involved in the way that the DNA repairs itself, has a very potent effect. But not everybody who carries that gene will end up with breast cancer or ovarian cancer. So there are lots of other genes that are present that moderate the effect of that particular base pair change. We also find genetic changes in tumors, and this is lung cancer. And here's the protein stained. You probably can't see that very well, but that's actually showing us that there's a translocation in this particular gene, which is the EGFR gene. And here you can see that there's the sequence that we get out of our more modern sequencing protocols. And this is one I like. I like pretty pictures, and I do like looking down microscopes, and I make no apologies for that. This is HER2. Now, many of you will have heard of that. HER2 is the gene that if it's present and turned on in the way that it is here, we would then give a breast cancer patient Herceptin, which actually revolutionized the care that we can give to those patients. So this particular protein is actually located on the cell membrane. So there's the nucleus there, and if you can see just round there with my shaky hand, you can see there's a dark circle around the nucleus, which is the edge of that particular cell. And if you can see here, the green dots are the centromeres, so they're the middle of the chromosomes. And the red dots are the HER2 gene. And what happens in this particular cancer is that you get lots of copies of that HER2 gene because they are present in lots of copies along the chromosome. And so those red dots are lots of copies lighting up with our fluorescent probe, and you have to count the number of red dots compared with the number of green dots in each cell. And because there's lots of the DNA segment there that codes for the protein, we see the protein overexpressed in the cancer. Now, the problem is that's nice and easy when you're dealing with one gene, but cancers are very complicated things, and they involve changes in multiple genes. And if you have to go through gene by gene, it's going to take you a very long while to do anything, and it's going to cost you quite a lot. So we have a problem in that we need to not just look at single genes. We need to look at multiple genes in very small amounts of tissue that come from patients. And this just gives you an idea of the genes that we know so far are implicated in lung cancer, in different types of lung cancer, but there are some that are present in all lung cancers. So ideally, what we want to be able to do is to look for multiple different genes at the same time. It saves time, it saves money, and it means that we can look at the interactions of those genes. And what we're starting to do now is to start to put together clinical trials where we'll take a piece of a lung cancer from a patient. We'll look with a technology that enables us to look at multiple genes at the same time. And based on what we find in that individual patient, we'll put them into different arms of a clinical trial. And it gets very, very complicated very quickly, because some of these genes, if you combine them together, mean that you can't give a drug that you would otherwise give. And other genes mean that you need to change therapy completely. So it's getting much more complicated to do these sort of things. And we used to have clinical trials where we had two arms: treatment, standard of care, other treatment that we were testing. And many of these trials now have 8 to 10 arms based on the molecular findings that we see in the tumors. So they become increasingly complicated to do, and of course increasingly complicated to explain to patients about what you're doing as well. Why aren't you getting the same drug as somebody else who you happen to meet in clinic? So it is going to be a challenge, as we learn more and more about this sort of technology, to be able to explain to patients why we're treating them the way we are. Now, the technology that we use to be able to look for these multiple different genes in one particular specimen is called targeted next-generation sequencing. So we've gone from doing single genes to doing multiple genes. And this is the best way that I can think to put it over. Those of you who are old like me in the audience will remember this nice fishing game, where you have a magnet and you can pull the fish out depending on pulling out the magnet. It's a bit like that when we do this sort of technology. We have a tag that enables us to be able to pull out of a soup that we've made of the DNA. We've chopped it all up, we've made a soup of it, and we're now pulling out specific genes using specific sequences and taking them away and sequencing them. That's not a very expensive technique, and in fact, it's now as cheap to do 50 genes with this technology as it was to do one gene mutation a few years ago. So all of a sudden, this multiple gene testing becomes extremely important. This is the way we used to do it many, many years ago. You used to sit down with your student with a see-through ruler, and you used to read this by putting the ruler across there and going, yeah, A, T, C, G. And the way you looked for mutations was where you had more than one band in each of those 4 lanes. That used to take ages. I used to spend hours with students doing this. Now we can do multiple genes, and this is a chip, and you can just see how many dots there are on there, sequentially, with the modern machines that we have. So it's incredibly fast, it's much cheaper, and it's much more accurate than it used to be, and it uses smaller amounts of starting material, so smaller and smaller samples, which is important because we're getting better at detecting cancer. Cancers are getting smaller, so we need to be able to use less tissue. Because we must be able to use most of it to tell us what the tumor is, because that still directs our therapy, but we are going to be able to take some of it now to be able to use in this technology to look at the genetics behind it. Now, that's okay if you know what the defects are that you're looking for, what the gene sequences are that have changed. But we know quite a lot already, but it is handy to know what you don't know. So the only way that we can look for new genes is to sequence the whole genome. We have the genome sequenced. We know what a normal genome should look like. So we now need to look at ways of looking at what the entirety of the genome is in a given tumor to be able to identify the new mutations that we've missed by taking a targeted approach. And the best way that I can explain this is it's like taking a book. And what we do effectively is we take your DNA sample and we rip it to shreds. We divide it into tiny portions. And we then try and stick it back together again, which sounds a bit mad, but actually it works extremely well. So imagine you took a Mary Berry recipe or something like that, ripped it all up, and then tried to stick all the sentences back together again in the right order. Now, if you don't have a template against which you can put that, it's going to take you forever, because you could end up making complete nonsense of the code. So that's why when we do this sort of technology, we say to patients, we need a sample of your germline, because that's your template. We also need a sample from your tumor. And what you do is to compare the different reads that you get out, because you just sequence small bits of this, a bit like taking that bit of paper and reading that bit. and we sequence them many, many times over and compare them with the original sentence. And you can see there that if you do it enough time, you're going to be very confident about the amount of this normal sequence that's there. Because when you look at a tumor, it's not just the mutated cells that are present in the tumor. You've also got normal cells in there, and you've got normal cells, the stromal cells that support the tumor cells in there as well. So you need to be absolutely confident that what you're reading is not normal, and it is consistently abnormal in some way or another. And I'll show you a couple of examples in a minute. Okay? Also, when you do it many times over, and that's called the depths of sequencing. You'll hear people talking about the depth of sequencing. You can work out what proportion of the DNA that you've sequenced is normal and what proportion is aberrant, or it may be aberrant in a different way from your original. And that tells you, or at least gives you some idea about how frequently the cells are mutated. Because what you need to do is to make a decision based on how many cells that you've got are having that mutation, whether that's a good target to use for your therapy. If you've only got 1 or 2 cells in the sample that are mutated, the chances are they're probably not the major driver of the tumor. So we're only really starting to get a handle on this, and there is so much work that now needs to be done in saying, where's our cutoff level before we say that is a good target for drug therapy? What proportion of the cells need to have a mutation? Does it make a difference? We don't know that. So we're going to have to do an awful lot more complicated clinical trials to answer those sort of questions. So the different types of change that you can have in DNA sequence are really sort of given here. A point mutation is literally just one base pair change. Okay? Now that can make a big difference if it encodes for a protein. It may introduce something that tells the protein that's it, stop, you're not having anything made after that point. It may be something that changes the way the protein interacts with something else in the cell, and the change in its binding site. So even a single point mutation can be very deleterious to the function of the protein. And a good example of that is the BRAF mutation that is very common in melanoma, that we now have very good targeted treatments for. And things like mutations in the EGFR receptor, which make you more susceptible to responding to a drug like Iressa for lung cancer. So just one change can make a big difference, but it depends on where that change is. Translocations are when you get a bit of the sentence that's chopped off and translocated somewhere else. Translocations usually involve 2 different chromosomes, so a bit of chromosome 1 can end up on a bit of chromosome 17, for example. They can be reciprocal, so it can be that they just switch those bits of information, just switch between the 2 chromosomes. They can also occur on the same chromosome, and you can sometimes get an inversion, and that's a very common mutation in some thyroid cancers, is you actually get a piece of chromosome that's turned on its head. You can also get copy number variation, which means you've got multiple copies of the same gene strung along on the line of DNA. And that's what you saw earlier when I showed you the HER2 positive breast cancer. Now, it used to be this was incredibly expensive to do and took a long time. But you have a thing called Moore's Law, which says things halve each year with the technology. And you can see just how rapidly the cost of sequencing the whole genome has gone down. And that means that things that we thought of, even in the early part of this century, would be years before they became cheap enough, have suddenly become something that it's possible to fund on the NHS. So you could have a whole sequence done for around about $10,000, and it's now coming down to around about $1,000 in this case. Okay? And in fact, you can have your stuff sequenced by just going commercially, taking a buccal swab and sending it off, and somebody will come back with your sequence. What you're supposed to do with that information, I'm not quite sure, but you can actually have that done, and many people do it. I don't think I really want to do it. I don't want to know everything about myself. It's bad enough knowing what I know. So this is just to introduce you to something that is starting up, and you'll certainly hear an awful lot about. This is David Cameron's big idea. Now he's back in power, he's certainly going to be pursuing this one. This is the 100,000 Genomes Project, which you'll see periodically, either with good press or with bad press, because it's quite a controversial project. The NHS is a brilliant organisation. It sees most of us from cradle to grave. It has massive amounts of data on what happens to us through our lives, and we don't really use it as a research resource. I've never really understood why we didn't do that, but there seems to be a complete different mindset between NHS patient care and researchers, and that's why we have things like our academic health science centres. We're trying to pull things together because there's a wealth of information, there's a wealth of tissue that's sitting in NHS stores, which would be incredibly useful if we could organise ourselves for projects like this. So the idea is to sequence 100,000 genomes. Okay? Now, that doesn't mean 100,000 patients, as you'll see a bit later. And that's to take the whole sequence and look at it and try and identify new mutations in rare disease and in cancer. So the aims of the project, of course, are to bring benefits to patients. We shouldn't be doing it otherwise. To create an ethical and transparent program based on consent. Because this involves your germline DNA, it is going to be controversial. So there's no point in us trying to do this in a silo. We need to be talking to people about it. We need your opinions. It needs to be a society decision on how we take the project forward. Obviously, to enable scientific discovery and medical insights, to kickstart the development of a UK genomics industry. Because we really do believe that the UK has something to offer here that many other places haven't got, because we have the NHS. So the idea is to sequence what they call trios for rare disease. So rare diseases on their own are rare, obviously, otherwise they wouldn't be called rare diseases. But actually, an awful lot of people have a rare disease. So when you look at the health burden to the economy, there's a lot of people out there who've got a rare disease who need help. And the idea is to sequence 15,000 patients. That's 45,000 genomes, because this is just taken from a germline DNA sample. But we want not just the patient, but also the patient's relatives as well, two of the patient's relatives, usually the biological parents. Now, those things are always sort of fraught with difficulty, because sometimes when you sequence the father's DNA, you find out he couldn't possibly be the father of the child. And there have been quite a few cases where they've had to stop studies like this, because it's a little bit difficult to have that conversation. So we just hope we get the right people in. And from cancer patients, we want blood and some form from the tumour. At the moment, we're having to work with frozen, but we hope we're going to be able to use what we normally use in pathology, which is formalin-fixed, paraffin-embedded material. That now looks quite a large number, and it is a large number. The trouble is that's multiple different types of cancer. So actually, an individual cancer may not have as much data as we would really like, because we do need large amounts of data, because you're all infinitely variable, and your tumours are all infinitely variable. If you just look around the audience, there are no two people in this audience who look alike, and there's certainly no two people who've got identical DNA, because I can't see any identical twins. And the aim is to provide a very large data, and this is where some of the ethics gets difficult, and a biorepository for genetic biomarker discovery and complementary omic studies. Now, to put that in some context, a lot of you will have seen the news recently about the new ovarian cancer test. That came as a result of a similar study. It was a different type of study called the UK CTOC study. And it just shows you how these large data sets and sample sets from patients can be incredibly valuable in looking for new biomarkers. Now, I can tell you this is not an easy experience. I've been working on it since August non-stop. If you do things with the NHS, they do tend to use an awful lot of paper, a hell of a lot of paper. So there's been an awful lot of form filling, but they finally announced in December, just before everybody went off on Christmas leave, a list of 11 centres that are going to participate in this project and really start to change the NHS. And we, of course, are one of them. We started off calling ourselves the West London Centre, and then we were told, no, you have to be in Imperial College Health Partners, and now they've gone back and said, no, actually, we want a geographic identity. So we are now the West London Centre. But you can see they're spread all across the country. There are a couple of holes, and they are going to look for 2 centres in Hull and Bradford and that sort of area of the north, and also Bristol, because there's 2 gaping holes because nobody applied to be a centre there. There are 3 in London, which will cover most of the London centres. So what will this project be able to do? Identify new causative genes for rare diseases, and you'll see at the end of the talk they've already done that for 2 patients in Newcastle, and they haven't even started the main project yet. So there's no doubt that we will achieve that. To identify new targets for cancer therapy. I can't stand here now and promise you that by the end of this project in 2017, we will have given you new cancer therapies, but we may have identified targets for them. Because actually taking the target through to having a drug that's effective and works in a patient population still will take us years to do that. But if we don't start somewhere, we're never going to have these new therapies. We will be able to investigate gene function, not just look at the sequence, because we're going to take what they call multiple omic samples. So we're going to take some serum, we're going to take some plasma from patients as well, and we will be able to use the tissue to understand how the DNA change translates into a change in protein function. It will generate huge amounts of data, and it will generate huge amounts of potential for research. The other thing that it is most likely to do is to generate new treatments or more targeted use of therapies we already have, which is the quicker option. We don't have to develop the new drugs. We don't have to go through the clinical trials to do them. We actually could use the drugs that we have much smarter. So instead of doing this, which is, to be honest, this is really what we do most of the time for cancer. Some of what we give you will hit the target, but not everything. We want to be doing this. And that should mean better responses, better outcomes for patients, and less toxicity, which actually will also save the health service money. Now, this is going to be a very long-term relationship with the donor who comes into this project. They're going to have to agree to receive feedback on findings related to their disease. But because we're also having to sequence your germline DNA to be able to see what's changed in the cancer patients, we are going to find things that maybe you don't want to know about. And it's a big decision as to whether you want to receive feedback on findings that might help your future healthcare or possibly those of your children. Now, we will also find things that we don't know what it means. And we won't know what it means until we've done an awful lot more research. But it might be something that we might need to feed back to you later. So we may have to reinterpret the data we've got, and we may need to contact you in the future. So this is a very different style of, it is research, but it's not research. It sort of bridges the two, which is what the NHS should do. But it will be a totally different mindset in the way that we do this project. The project will definitely reshape how we run the NHS. It is going to drive change in different places in the NHS. We need to collect these samples in a slightly different way from the way that we normally collect samples for pathology. It is definitely going to change pathology. That is not going to be easy. Changing anything in the NHS is an absolute nightmare. But it's going to have to change because we need to be able to deliver this. It raises very important ethical issues, and I'll highlight some of those in a moment. It should lead to much more research, better treatments, and better outcomes for patients, and that's what the NHS has always been about. Will it lead to better health economics? I think we need to be really careful about adopting technologies because it's sexy. We need to make sure that sequencing the whole of your genome at the cost of £1,000 is actually going to benefit and better treatment for you. Maybe it doesn't. We'd like to think it is because we all like technology to work. But, you know, we have a restricted amount of money we can spend on healthcare, and we need to spend it wisely. We have the technology to do this. We have it now. But what are the ethical and the legal issues? Some of these are the ownership of data. Who owns the data once we've generated that sequence? Does it belong to the patient? Does it belong to the people who've generated the data? How can that data be used? How can we exploit those results? We are not going to be able to make new drugs, new therapies in universities. It requires so much money, it's going to have to be done by industry. There has to be industrial involvement in this. That makes people nervous because there are privacy issues around somebody getting hold of your genome. And we'll come back to this again at the end. We also have an ethical dilemma in what should we feed back to you? When should we feed it back to you? How should we feed it back to you? How do you want this fed back to you? What we think as professionals may not be what everybody else thinks. So we're going to have to actually talk to people. That'll be a bit of change for the medical profession, but we are going to have to communicate better. How is this going to affect society? It has the potential to lead to genetic discrimination. We can discriminate on race because it's easy to do that. We can see the phenotype. Once we've got the genetic ability to be able to sequence people's genomes, we could possibly end up with some form of genetic discrimination. Obviously, the first thing people think about is insurance. Some people have already suggested, when I floated this to patient groups, well, this would mean that we could select embryos that don't have the defect that causes our disease. Now, that might be beneficial in some diseases, but in others, actually, when you inherit that gene, you also inherit a suite of other genes that affect your health generally. And there are certainly some groups of patients that inherit a gene that gives them a disease, but also seem to inherit something that protects them from getting cancer. So if we start fiddling with our own gene pools, there are all sorts of societal implications that can come from that. And we need to discuss this. We need to be open and transparent about it. How much is it going to cost? I think we've already covered. I think, for me, the most critical point of this is, does it add to conventional medicine? Does it give us something more for the cost? And who's going to pay for this test? Does it mean the oncologist is going to pay for it? Does it mean it's paid for by pathology? Because that's what often stops technologies being adopted, is who actually is going to pay for the test within the different budgets within the NHS. Now, ethics, law, and societal issues are incredibly difficult in medical research because they do involve humans and people have different opinions. But generally, the way we approach them is we have to look at the risks and benefits to society compared with the risks and benefits to the individual. Most of the time in medicine, the weight is definitely down on the individual. It's better for the patient to be treated this way individually. But sometimes, actually releasing a little bit of that autonomy will enable better things to happen for society. So at the moment, you can take DNA for diagnostic purposes without consent. But it is illegal to take DNA for research as part of the HTA or paternity testing without the donor's consent. This consent has got to be informed, and there's a big issues around informed consent, which I'll go on to on the next slide. Research projects that use material and data from this have got to have undergone a review to ensure that the use is appropriate and covered by the consent. So if there are opt-out clauses in the consent, you can't do that research using those samples. And the samples have got to be anonymized to researchers. You may want to bring back the clinical data and cross it with those samples, but to the individuals actually carrying out those tests, they need to be anonymized. So there's a whole load of issues that you've got to go around anonymizing samples when you do this sort of research. Now, the problem we have with informed consent is a bit like if you go into a nice restaurant. If you've got about 6 things to choose from, it's fairly easy. If they give you a 20-page menu, you sit there going, oh, no, I'm not sure. Oh, well, no, I think no, hang on. Oh, that looks nice. And it confuses you. And we have a problem with informed consent here because I can tell you all the ins and outs of the genetic tests and all the things it might do and the percentage chances and all the rest of it. Does that help you make a decision whether you want to go into a study like this or not? I don't think it does. And most of the patients I've actually spoken to said they'd much rather have a very broad generic consent, but a lot of information available for them to look at if they wanted to. And they trust us to make the right decisions on their behalf. And we have the same issue when it comes back to feeding information back to patients. If I feed you lots of information and don't give you context, you're going to worry about it. It's like taking all the statistics you hear on the news about don't eat this food, don't eat that food. Some people will get really stressed out by that. So we have to think through very carefully, and this is where we need a lot of public involvement. How much do you want to know? How much do you want to be able to find out about if you want to, and not everybody does? And how much information do you want back, and how would you like us to talk to you about it? And DNA is no good on its own. It needs context. It needs data around it. You need data on the patient. Not who they are, but how old are they? Are they male or are they female? You need data on the disease. What type of pathology is it? What does it look like if it's a rare disease? What do the scans look like? So you can tie that genetics into what you see in the patient or in the pathology under a microscope. We also, if we're going to find new treatments, we also need data on treatment and outcome. What happens to that patient? Now, you'd think this would be quite easy in the NHS, but the NHS IT systems don't talk to each other. So we have a real problem of trying to pull the IT systems together, which I think is the biggest challenge of this project. And outcome is particularly difficult. Cancer patients are living longer, and London is terrible. You set up a trial in London. Trying to do it with elderly patients is no good. Most of them live on the south coast. So if you start something like this with somebody in their 50s and 60s, they can have moved several different hospitals by the time they get to their 80s. So we need to find a way of being able to link the data that we have, and we have national cancer registries now that will be really useful for doing this. But sharing data within a large organisation poses huge problems. So there are big issues here that we have to think about. And the problem that you have as individuals is the larger the data set we have on you, the more likely it is that we could actually identify you. The more you know, the snippets of information, the more you can piece together. So that's what most people are worried about. But actually, are we so right to be concerned about that aspect of these sort of projects? You see, I bet many of you use Google, don't you? Who doesn't use Google? Everybody, you use Google. Google amasses huge amounts of information on who you are. Not what your DNA says, which actually doesn't tell you very much about what you're thinking about and who you associate with and what your politics is. But Google can do all of that just by looking at your searches. Have they asked us for consent? No, they haven't. So why are we so hung up about somebody having our genetic information when it really doesn't tell you that much about you, the individual, and how you think, who your friends are, who your family is? We also use Twitter, and we have had an inquiry from somebody who wants to put the whole of their genome sequence on their Facebook page. Might sink Facebook's servers if a lot of people try to do that. But literally, some people are willing to be that open. They are willing to have the whole of their genome on there. And you will be able to have all of what they call the BAM files, which is the data that comes back. And I have been reliably informed, although I wouldn't know where to start, but my son might, that you can just load them up into various things that exist on the cloud, and that will give you a readout of what that data means. You won't have anybody to put it into any context, because if you took it to your doctor now, they just freak out and say, I don't know what the hell that is. But you could do that. So there are huge issues around this that we need to be thinking of as a society. We can't get away from it. These developments are going to affect us all, hopefully in a good way. This project actively encourages patient and public involvement because it is so revolutionary. And if we don't take you with us, how can we move the NHS forward? We are all patients of the NHS, at least at some point in our lives. Although I must admit, I avoid doctors whenever. I work with them. I know what they're like. It's our NHS. We own it. We run it. So we have a right to express an opinion over this. This is our money that's being used to fund it. So I would encourage you to look out for opportunities to be involved with your local genome medicine centres, wherever you're living, as this project evolves. Because if you don't say anything, you're going to lose the option to do so. And you might find that decisions are being made that you don't really like. I think it is really time that we started having grown-up conversations with people who use the NHS and start to talk about, this is the way medicine is going. Is this the way you want it to go? Should we rein back on this? Are we going too quickly? If we don't take society with us, we could end up with an awful lot of problems. Just to end on a positive note, this was a pilot study that was run in front of the main study to try and find any problems. And even in that pilot study, and the patients who actually gave their DNA for this have been quite open. They're on the internet, you know who these people are. But they are absolutely delighted because they have the first 2 new diagnoses. And those patients are over the moon because they have a label to put on their disease, which is incredibly important psychologically. Now, if you want to know more, go and have a look at the 100,000 Genomes Project website. We will get our own pages up in Imperial, as will all the other GMCs, to point you in the direction of more information. And I would recommend watching the video here that's been done by Vivian Parry, who explains about genomics in a way that I probably never can. As a journalist, she's much better at doing this. And if you watch that, I think you'll find that very interesting. It may raise an awful lot of questions. And you can, of course, question the website as well. So I would encourage you to get involved in this project. Not just because you might have cancer, but because you're citizens. And we have a right to decide where we're going with huge projects that may affect society like this. Thank you very much.